Hello, everyone. I'm Alex Lee, General Manager of iCatcher. Today, very glad to have all of everyone to join in the webinar today. Our webinar today is going to share iCatcher novel solution for cfDNA and cfRNA purification from liquid biopsy. We very glad to have four speakers to join in the webinar today. First, we will invite Dr. Hsiao to have some presentation about his cancer research, mutation profiling in cancer through FFP and cfDNA samples. Then we will have Dr. Rudolf Walser to present catching rare genes from liquid biopsy. And finally, we will have two users' feedback. One is Tonia, another is Tzu-Hung. So for our webinar today, we will take around 1.5 hours totally. If we have any question, if you have any question, we have a QA section finally. So please ask. You can ask question in the chat box, or you can write your question in the Q&A section. Okay, so let's move to the first section of this webinar. Let's welcome Dr. Hsiao to share mutation profiling in cancer through FFP and cfDNA samples.
Do you see my screen?
Yes.
Yes.
Okay, I will thank you, Alex, the invite. I have the opportunity to give a talk to introduce my study in prostate cancer and the thymoma through FFP and the cfDNA. I will talk about some of my result of my research and also mention about how I use the iCatcher to extract the DNA and to help me and my lab to perform the research. So first one, we will talk about the mCRPC, the metastasis castration-resistant prostate cancer. As we all know, the prostate cancer is addicted on the androgen pathway. So the initial treatment of prostate cancer is about the castration. But after for a while of treatment, then the prostate cancer will resist castration resistant and come back with the metastasis. And at that time, that we will use another treatment we call the abiraterone and the enzalutamide is the very high sensitive the castration for inhibit the androgen, and the two drug is the less than 9 drug for treat prostate cancer with metastasis. However, about the half year or 1 year treatment, then the prostate cancer will progress again. And now we want to try to find out the prostate cancer have another addictive oncogene to promote the resistance and the drug resistance. And the recent study shows the DNA repair gene mutation may be one of the keys. And the recent study shows about the 12% of germline mutation was found in prostate cancer, and also in other study they find about 11% mutation in about the 700 mCRPC patients. Okay, so now the study is focused on DNA repair gene, and some research find out about 3% of patients have the somatic mutation in DNA repair genes. And why we focus on the DNA repair gene, because the drug or the PPAR inhibitor is very useful for patients with the DNA repair gene. And by the most the literature is focused on the Caucasian and the Black people. The mutation profile of Asia, Taiwanese, and the Han Chinese is still unclear. So, for example, one study in Japan, they find about 2.9% germline mutation of DNA repair gene in their prostate cancer patient, but in Chinese research, they find up to the 10% mutation in their patients. But we all the Asian. So the result is still the controversial and unclear. So in my study, we want to try to find out how many somatic mutation in the metastasis castration-resistant prostate cancers they under the treatment of abiraterone and enzalutamide. And this project is cooperated with Dr. Oh, he is a urologist, and also Professor Hsiao, he is the epidemiologist. Okay, so when we try to do this research, we only can collect a sample from the cfDNA. So we need to use the cfDNA to detect the mutation on those patients. So we try to use the iCatcher to perform the DNA extraction. Now, actually, why I use the iCatcher, because actually it's their design, all the reagent is put on a line, and it's easy to use and can avoid the contamination. Actually, the amount of DNA in cfDNA is very, very limited. And so we want to avoid the contamination as we can. We also compare, actually we have the quizivny, so we also try a comparison with the Catch-Gene iCatcher with the quizivny quizivny to compare their extraction year and the purity between the cfDNA. So we recruit about 30 people. Actually, they are normal people, and we collect everyone got a 4 milliliter plasma through packaging the cfDNA specific tube. Then we compare the result, and the result shows actually the iCatcher have the more higher year compared to Quizivny quizivny. Make a show conclusion. Actually Quizivny quizivny is good. Yeah, but iCatcher is more better for the year. We can show this is the same sample, and this the electrophoresis and the iCatcher have the more broad and a higher peak, and both of them have the long fragment that shows actually the extraction is pretty good, but the iCatcher, the year of the iCatcher is more higher. And we also make a summary about the total DNA amount of the cfDNA. Most of the samples was more than 20 nanogram. And I think 20 nanogram is the minimum requirement for my lab to perform the research, because, for example, we need a 10 nanogram DNA for comprehensive cancer panel, or we want to do the whole genome sequencing of cfDNA, we need a 10 nanogram. If we want to do some research on methylation capture, we need a 15 gram. And we also need some validation for PCR and TR cloning for Sanger, or maybe we need a 10 nanogram cfDNA for digital PCR. So I think the minimum requirement for my protocol is 20 nanogram. And for this little survey, all of the result of from iCatcher is meet the requirement of my research protocol. And this is the data provided from the iCatcher, and you can see the repeatability is still good for the three biological repeat. The result of UFO is similar, and the year is quite stable and good. And we also try to use the iCatcher to extract the FFP samples, and you can show for FFP DNA extraction from the UC. And we use the 5 slides. Every slide got the 10 micrometers. And the year, both the year is quite good, but oh, sorry, I made a mix. This is iCatcher. I make a typo. This is the iCatcher, and this is the Quizivny, and the iCatcher is more higher than the Quizivny FFP extract kit. And we also use the iCatcher to extract the RNA, and the year is all pretty good. And we choose 3 samples to perform the qPCR. Even if it's FFP RNA, the fragment is short, and as we know, the RNA of FFP is not good enough, but we still got to perform the qPCR and the GAP-DH, the delta CT of GAP-DH about 27, 26, the performance is quite good. enough to perform the qPCR through the FFP RNA extraction. Okay, back to my study. In short time. We performed the comprehensive cancer panel on 78 MCRPC patients, and we find out about 11 pathogenic and the pathogenic-like variant, and all of them are known the oncogene. For example, we got the 2 AR gene mutation, 1 TP53, and 1 AKT, HRAS, P10, BRAF. Yeah, so we got the 10 mutation genes. And we performed the survival analysis. So we find under the treatment of abiraterone and enzalutamide, when the patient with the somatic pathogenic mutation, they got the worst survival curve. We will consider abiraterone alone or enzalutamide alone, they also shows the worst survival too. So the data shows the other mutation. If we find the other mutation, DNA repair gene mutation or oncogene mutation on the cfDNA, and the patient tend to get the drug resistance of these 2 treatments. Then we do another study because we know that the treatment will decrease the PSA, and the patient is in a stable status, then the tumor will progress again. So we collect the 3 time points. One is stable, one is progress 1, and one is progress 2, and extract the cfDNA and perform the comprehensive cancer panel to see what's the difference of the mutation profile in these 2 time points. And the result shows actually we did not observe the difference between the mutation burdens. They are all similar, but we can find the number of pathogenic somatic variant will increase. This is the blue one is the consistent progress, and when patient has a stable disease, the number of somatic variant is among the 15. Then when they move to the progress status, then the number of pathogenic variant will increase, and then through the status of progress 2, the increase more up to 22, and the comparison shows the statistical significance. Yeah, and we also include more. The one is just the pathogenic variant, and this is include the pathogenic and the pathogenic-like variant. We observe the similar, and when the disease progress, the accumulated pathogenic and pathogenic-like variant will increase. So not only the number of the pathogenic variant increased, we also observe the allele frequency in the detect. The sample which carry the mutation will also increase. So we can show this is the allele frequency. So we just pick up the patient with the BRCA2 mutation. You can show for patient with the consistent progression, actually we can detect about more than 2% allele frequency of BRCA mutation in their cfDNA. But for the patient, initially it's stable, then progress, the MF of BRCA2 mutation will increase. Yeah, and we also find actually for the stable patient with stable, actually in the beginning this treatment response, and the blue one is initially treatment resistant, and we can observe the ATR mutation allele frequency is more higher in the treatment resistant patient compared to treatment response patients, and also in P10, and the check-do is changed. So we can observe those mutation. We can detect those mutation in the patient's cfDNA, and then they can respond to their treatment status. Okay, this is in our lab we perform research on the cfDNA. Now we will jump to another topic. We use the FFP sample on thymoma to do the mutation profiles, and we find that actually for Taiwanese thymoma, we got a distinct mutation profiles. And thymoma is a thymus, the cancer, and the incidence of thymoma is about 0.13 to 0.15 in the 10,000 people in Western. But in Taiwan, we up to 1.61 per 10,000 people for men and 1.33 per 10,000. 100,000 people in Taiwan. So the incidence rate is more higher than Western. So we hypothesize actually for Taiwanese thymoma, we will get the different mutation profiles. So we performed a study from the FFP samples. We collect the 47 thymoma FFP sample and extract DNA from iCatcher and to observe the mutation profile. And the major mutation of the thymoma is about the F81 genes. And we also observe the DNA mutation gene ATR and the MASH6 in the 47 samples of FFP. And compare the onset age, we can find, for example, MSH6 patient. The patient with the MSH6 mutation is more younger than patient with the wild type. And we also find another genes span the CD276XX that carry in the younger patients. And also we also find the PD1A3, VSIR, and the DMT3A the mutation is detected in the elder patients. Then we use our result to perform the mutation signature, and we find that the major is about the SBS5 and the SBF26 mutation signature. And actually the signature is about the DNA repair gene. Again, the DNA is appear again. So we go back to see the DNA repair gene. Actually, we also got the evidence. We got the MSH6 mutation. We got the POLE mutation. And for MMR and for NER, we got the LIG3 mutation, ERCC2, ERCC4. And we observe the mutation. We observe the mutation signature and find it's associated to DNA repair gene. And we also observe the DNA repair gene mutation in DNA level. Then we also have the RNA-seq. So we just look at the gene set enrichment. We also find the DNA repair gene. For this one, the reactor mismatch repair is also enriched in those sample carrier DNA repair gene mutations. And finally, we compare the mutation profiles between Taiwanese and Caucasian. And this is data from TCGA. Actually, for the thymoma, everyone knows that most of the Caucasian thymoma carrier GTF2I mutation. Yeah, you know that you can see that got the last people carry this mutation. However, we can find no patients carry this mutation, and otherwise you can see the picture. Actually, the mutation profile between the Western and the Taiwanese is quite different. And we also perform the pathway analysis. The data shows actually for Taiwanese patients, we got more enrichment on, again, it's mismatched DNA repair genes. And we compare the mutation signature. Actually, we also observe the SBS26 is more higher in Taiwanese patient compared to TCGS data. And also the enrichment, the analysis also support the result. So make a conclusion, actually, we got a different mutation profile in Taiwanese and the Caucasian in the thymoma. And in Taiwanese, maybe the DNA repair gene mutation played a more important role than the thymoma patient in Caucasian. Okay, this is my brief introduce how I use the iCatcher to perform my research. Okay, the most important things, if you just only have cfDNA or FFP, actually it's hard. It's very hard and it's very important to extract enough amount of DNA or RNA from those samples because they are not frozen samples. It's hard to extract. So this is a critical point to get enough amount because we need to do sequencing, we need to do validation, maybe we want to try the methylation, do that's a bunch of experiment. So the critical point is got enough amount of DNA RNA. And the two studies we use the iCatcher from the cfDNA to extract the sample of our lab, cfDNA and FP, and got a high yield and high purity DNA, and to perform those results I show you. Okay, thank you for your listening. Thank you.
Okay, thank you very much for Dr. Hsiao's presentation. And I hope you really enjoy of using iCatcher.
Sure, good.
Thank you. And for the next presentation, we will have Dr. Rudy, and his topic of presentation is catching rare genes from liquid biopsy. And Dr. Rudolf Walser is a molecular biologist, and he is also our European representative. Okay, let's welcome Dr. Rudolf Walser.
Okay, dear colleagues, I'm happy to introduce you today to the iCatcher system, which is a novel purification solution for circulating cell-free DNA. As we already heard from Dr. Hsiao, with his impressive examples, it's important to have the right tools to get access and do the efforts that it's needed to do in cancer research, for example. And as Alex said as well, my name is Rudolf Walser. I'm the representative for CatchGene in Europe, in Middle East, and in America. First, before we go to the liquid biopsy part, we have a look to the golden standard, which is used in diagnostics at the moment. So it's tissue biopsy. Tissue biopsy is great to get access to the targets, but it has some limitations. So the problem is always the sampling. If we have tissues which are hidden in the body or which are very small and not easy to reach, then it's problematic to find the right samples. As well, the timing, especially for patients which are under treatment, they are not easily can be treated by these little operations that this is always an operation or something which is very harmful for the body of them. And it cannot be done frequently. Okay, but nevertheless, the scientists are very intelligent normally, and so they found novel solution for detection of parts in the body which are not easy to access. And liquid biopsy is, for example, a very common novel solution for cancer detection. The benefit is it's non-invasive, so we can just collect it from any body fluid, for example, from blood sample, and the monitoring can be done as many times as necessary because blood sampling is done in the world millions times a day, and it's not very invasive to patients which are under treatment. The parts that we are looking for are normally, especially if we talk about cancer detection, we look for circulating cell-free nucleic acids. Every one of us has circulating cell-free DNA in the system of the body fluids. And due to apoptosis and necrosis, genomic DNA is released into the circulating system and degraded into smaller fragments. This smaller fragments is called circulating free nucleic acids. Before, people think that circulating cell-free DNA is just junk or okay, good for recycling of the components. But during the past 2 decades, scientists realized that it's not only normal cells that will release their DNA to the bloodstream, it's also cancer cells, it's embryo cells, even transplanted organs just contribute their nucleic acids to the bloodstream. So if we are able to catch enough and pure circulating cell-free DNA, we are able to detect and monitor if there is any tumor inside our body. This is also very nice for fetal DNA investigation or to check the situation of transplanted organs. But as every nice method, there is also opposite part of the medal. We have heard from Dr. Hsiao already that the challenge is in the purification of the cell-free DNA, because cell-free DNA is very rare. So only 1/20 or 1/10,000 of genomic DNA is cell-free DNA. So it's only 1 to 20 nanogram in 1 milliliter of blood that we have this target that we are looking for. On the other hand, it's very small. It's only 150 to 200 base pairs, which is about 1/1000th of the genomic DNA, the mid-size of the genomic DNA. And these 2 challenges, they lead to the situation that most of the instruments on the market, which are doing nucleic acid extractions, are failing for this kind of application, because most of them are still based on magnetic beads, and they are also limited, and this is the most striking factor, to 200 to 400 microliters of sample input, which is way too less. So what we heard already also from Dr. Hsiao, from his experiences, and they are good. These kind of extractors are good for genomic DNA extraction, for genetic analysis, or for viral nucleic acid extraction, for the disease detection. So therefore, they are really good and easy to handle. But if we go to this more hidden and secret part of cfDNA, these instruments are not really suitable. So what did we do? We thought about a solution. Here we are. The iCatcher was born for precision medicine. We have combined 4 important features to raise a powerful purification technology for circulating cell-free DNA. First, it's membrane column-based. So this works like a fisher net, and we can take out as much as possible of nucleic acids from the sample. Second, we use the wash-away method. It's like shower. Like shower means we just pour away the impurities, and the impurities are always a big problem in the downstream applications, especially if we only have a little bit of yield in the extraction. Therefore, we also looked at the third point. We are able now to use about 4,000 microliters of sample input, which is 20 times higher than the normal extractors on the market. And to be secure about cross-contamination, we choose the technology 1 sample in 1 line. Now, why can we get higher yield with the iCatcher? How can we capture more? Other systems like magnetic bead they work like, let me compare it to a fishing. We are fishing in a pond, and we use a rod. And yeah, we are able, if the pond is full of fish, then we are able to find and catch a few of these fish. But if we are able to use a net, a fishing net, which is comparable to a membrane column-based technology, then we are able to take out almost everything which is living in this pond. So about 90% or more we can catch. And this is the same principle that we use here in this case. And the recovery rate is about 300% higher than with magnetic bead systems. The next problematic thing, which is at least as important as the yield, is the purity. And here the magnetic beads have the most problematic part, because when you do the extraction by magnetic beads, you always have some interstitial residual of liquid between the beads. And these are carrying all the inhibitors with, and then you go to the next cleaning step and you still have this interstitial liquid, and you cannot get rid of that. With the iCatcher, with the wash away method, we just pour the whole thing through the net or to the membrane, and we can wash away the inhibitors by this way almost 100%. And this leads to very nice CT values when we do real-time PCR. So the CT values are 10 times better, or it means 10 CT better than if we work the same sample with magnetic bead technology. Yeah. And to combine all these things to a very nice system, the CAP technology was discovered. CAP means coated membrane, anti-gravity, porous membrane, and positive pressure. So the coated membrane, anti-gravity, and porous membrane, this is something other column-based systems also may use for them. But the positive pressure is one of the differences, because in comparison to other column-based systems, we are not using vacuum. And vacuum is always combined with air. Air is combined with turbulences, and turbulences are a nightmare for cross-contamination. In our case, we just pour the liquid through the membrane without air turbulences. So the cross-contamination risk is much, much less, or is almost nothing. Combined with the large volume input and the one sample in one line, it's a perfect dedicated technology to catch rare components, rare genes, rare cfDNA, cfRNA from the sample, and it's very clean. And in the end, it's that what we are looking for when we are doing the downstream applications. So Dr. Hsiao showed us a few nice examples already. And yeah, this is what we found during the past years, that this is really the fact that these components that we put together are the right way to this, yeah, let's say, field of doing extraction from liquid biopsy samples. Yeah, and now our general manager, Alex Lee, will have the chance to show us the instrument working, and that we get an idea about how the instrument is functioned. And let's start with this presentation, Alex. Thank you.
Okay. Hello, everyone. So thanks again for everyone to join in the webinar today. Unfortunately, it's still in virtual mode. So everyone online cannot touch the real system. But today, besides only presentation, we prepare some demonstration of the real system, real iCatcher system. Maybe you cannot touch it, but this chance will let you to see the movement of a real system, real iCatcher system. Okay, this is the iCatcher system. Actually, it's a benchtop system. You can see very compact in size. And from the front view, we design a hidden built-in touch screen. Here is the screen. You can just push and open a small door. Then here is a 7-inch built-in touch screen. To turn on the system, it's very simple. Just press the power here. Then once you press the power, the system will initialize by itself and do the self-calculation. That means it will go to the 0 point of each axis, like y-axis, x-axis, and the syringe axis, and do the calibration every time. So it's very precise. To that, the system become very precise. Now it's initializing and also doing some check action to make sure there isn't any syringe or tip. There isn't any tip on the syringe. Okay, and once the initialize complete, the system will turn off the light. That means initialize already down. And we can see this, the home screen of the system, maybe a little bit small, but once it's show enter, we can enter into the screen, enter into the system. And all the icons is very geographic. We can easily understand the meaning. And let me turn on the LED light. Okay, and first of all, let me introduce the inner part of our iCatcher system. Here, actually, I think everyone already used to have some, for automation, I think everyone used to see some syringe or pipetting tip here. But actually for iCatcher, we use a disposable syringe instead of a fixed syringe. So that's quite different part of iCatcher system, because iCatcher system was born for liquid biopsy, and we want to handle very large sample volume. So based on this design, we can handle up to 4 milliliter serum plasma sample. And it's very easy to just push the syringe into and complete the setup. And also for example, iCatcher system, we use a special sample tube, which is similar to 40 milliliter vacuum tube. But actually, the difference between iCatcher with other system is it can handle very large volume. Like right now, there are 4 milliliter sample inside of the sample tube. Okay, just simply place it here. And there's another quite different part is we use pre-filled cartridge. And actually in very big size. This is my hand. You can compare it to my hand. All the reagent for 4 milliliter plasma sample already pre-filled into the cartridge. So if we want to set up the work table, we don't need to drain or aliquot the buffer. And one sample for one cartridge for one sample. It's very easy to set up the work table. We just need to tear this foil and put it into the system, like in this way, insert into the system. And then we can start the system. By the way, for the operation screen, we just choose proper protocol, and we can select different elution volume here. And also, the system the built-in touch screen also show the illustration of how to set up the work table step by step. Like I mentioned, first of all, put the syringe inside. Then put the elution tube over there. Then put the cartridge. Put the column tip set, and the ethanol tube and sample tube. After we done all the setup, there is an icon shows go will pop out. Then we can close the door and click icon. The system will do the purification automatically. Then what should we do? We can have a cup of coffee. Oh, okay. Just kidding. I'm back. Yes. For automation, actually, it's a way to help automate all because purification procedure is tedious. And in our system, actually, it's born for liquid biopsy, so it can handle up to 4 milliliter sample. But beside liquid biopsy, actually, we also can do FFPE. We also can do microRNA purification. And all these protocols are built in and can be done in the same system. Just use different kit. Now the system is purification. And another special part on our system is we use transparent screen window. At the very beginning, when the customer buy the system, normally they sit and see what the system is doing. They're curious about what happened in the system. Then for a while, the customer start to back to their seat and just hear the sound of the system. One of our customers told us it's kind of pressure relief, the sounds. Let them feel somebody is working for them. Okay, so it's a very easy to handle 4 milliliter liquid biopsy.
Okay, great.
Okay, and finally, let's wait for a while because later I will show you. Finally, it will elute in 1.5 milliliter envelope, which is compatible with most liquid handling systems. So it's a very easy to connect to your liquid handling system. Okay, and by the way, for different protocol, actually it's have different running time. For example, currently we have protocols for viral DNA, RNA, which is only 30 minutes. We also have protocols for large volume liquid biopsy, which takes longer time because 4 milliliter sample volume is much larger than 0.2 milliliter. It's 20 times. But our running time on iCatcher system only takes 2.5 hours, which means 150 minutes. Then it's fully automated from lysis, binding, wash, dry, then elution. Okay. All the things we need to do is waiting. And finally, we can see the elution too.
And having a coffee.
Ah, yes. That's right. Having my coffee. Okay. And once the system complete, we can hear a beeper sound. The robotic arm will back to the 0 position. Then we can open the lid, close the cap. The elution tube is in the inner part of the system, which means no action will cross the elution tube. So it also can make sure without any droplet into the elution tube. And finally, we can get minimum 30 microliter eluate into 1.5 milliliter Emmendorf. So which is very high quality, high yield cfDNA or microRNA or FFP DNA RNA. Okay, so hope this demonstration help you at least to see the real iCatcher system, not only photos or only videos. Okay, yeah. Thank you. So let's transfer the transfer back to Rudolf's presentation. Okay.
Okay, great. So can everybody see my screen again?
Yes.
Yes?
Yes.
Okay. So back from the work table to something you already have mentioned. The iCatcher is not only suitable for the cfDNA, it's a full capacity extraction machine for all kind of sample type. So we can extract, as you said, DNA, RNA, total RNA, and these from very various sample types. These are just examples here that we can just use, and there are many more. But our expertise, which is making the difference to our system, is the circulating nucleic acid extraction part. And to summarize why it's our dedicated field, it's just a very quick overview once more. It's the sample volume. Alex emphasized that several times. So we have a full range of 250 to 4,000 microliters of sample volume. We have a wide range of application, as we heard, and we have a high quality and purity of the eluates that we create with the instrument. And especially when it's rare and small, it's very important that the quality of the eluate is utmost high. So therefore, if you go in the field where this is important, then you should consider to have a look to the iCatcher. We always have good connections to let's say all detection methods in the downstream application field. So NGS, digital PCR, qPCR, mass spectrometry, and methylation methods are just examples for what you can do with our extracted nucleic acids. And now I'm really finished with my presentation. I want to thank you for your audience and your patience with the technical little issues that we have. And yeah, let's work together to let cancer become a chronic disease. Let's catch rare genes from liquid biopsy by using systems like the iCatcher. I'm Rudolf Walser, and here is my email address so that you can easily get access to me after this presentation and session. Thanks again.
Okay, thank you very much for Dr. Rudolph's presentation. Hope everyone enjoy it. It's very clear. And now, okay, we will shift to another session. Tonia. Tonia is a senior manager of R&D Center of Genomic Bioscience and Technology Cooperation in Taiwan. Genomics is the biggest NGS service company in Taiwan, and they're already using iCatcher for around 1 year, and already did lots of different samples. Today we are glad to have Tonia to share cfDNA for NGS cancer research about genomics company. Okay, Tonia, welcome you to share your presentation.
Hello.
Hello, Tonia. Yes, we can hear you.
Okay. Thanks for Alice's introduction. Hi, I'm Tonia, a senior manager of R&D Center of Genomics Biotech Company. Okay. Today, I want to share two applications of cfDNA genetic testing in cancer research. The first one was cfDNA EGFR mutation detection via digital PCR for lung cancer. The second was cfDNA whole exome sequencing via exome capture for pancreatic cancer. Okay. Before genetic testing, before genetic testing, cfDNA extraction was basic but important. First, must use proper blood collection tube, such as BD strip tube. It was very important because it can inhibit the white blood cell release genomic DNA into plasma. The second, use two-step centrifuge method to separate plasma and avoid cell debris into the plasma, avoid cell debris contamination. And third was iteration. In past year, we use manual kits such as Cryogen kit to iterate cfDNA but it is very time consuming. Consider cfDNA sample increase dramatically. We change iteration method from manual iteration to iCatcher. In recent 3 years, we have finished at least 5,000 cfDNA iteration. iCatcher makes cfDNA iteration easy and simple and robustically. The last one was cfDNA quality check, checked by a high sensitivity fragment analyzer. About cfDNA quality check, we need to consider two indicators. The one is cfDNA yield. The second was cfDNA fragment proportion. Here was the example of QC report for cfDNA for whole exome sequence. For the WES library construction, the minimum need 20 nanogram cfDNA, and the fragment proportion is the minimum need greater than 40%, and avoid large fragment DNA contamination. How to distinguish good or bad cfDNA quality? Here are the examples for cfDNA quality profile. The upper figure is the good pattern. We can clearly see the nucleosomer, monomer, dimer, trimer, and tetramer, and without detect any large fragment genomic DNA. This is a good pattern of cfDNA. The lower figure is GDNA contamination pattern. Also, we can clearly see the nucleosomer, monomer, dimers, trimer, tetramer, but we can detect the large fragment genomic DNA contamination. The genomic DNA contamination will affect genetic testing precision because we cannot clearly distinguish the genetic variant information come from cfDNA or genomic DNA. About cfDNA EGFR mutation detection on digital PCR, the cfDNA iteration was critical step. iCatcher helped to achieve robustity and quality and quantity. And after cfDNA iteration, we apply the cfDNA and the reaction into the chip, and it will embed in synthesis the droplet. After the droplet synthesis, we perform the immersion PCR to amplify the signal, and then we detect the fluorescence signal in the sense instrument. We use the software to analyze the signal, so we can easily to detect the mutation signal in here. The mutation sensitivity of this assay was 0.1%. Good cfDNA quality and quantity will increase genetic testing success rate. About cfDNA whole exome sequence, here was the workflow. After the cfDNA iteration and the quality check, we can directly put it into library construction without further physical or enzymatic fragmentation. After the library construction and the quality check, we use NovaSeq to get the sequence profile. Bioinformatics will evaluate the data quality via many QC metrics, such as Q30, duplication rate, mapping rate, mean coverage, target rate, and variant call precision rate. Here was the cfDNA reference sample WS data. Based on the Q30 score and the average quality score, we can see it shows sequence quality was very good. Behind sequence quality, cfDNA WS variant code precision were also evaluated. The real data and the expected data were consistent, and the precision score was greater than 0.95. The last one slide, I give a brief conclusion. cfDNA quality and quantity were critical points for genetic testing precision. iCatcher makes cfDNA extraction easy, simple, and robust. cfDNA reference material help to validate and monitor the data precision of genetic testing. Thank you for your listening. If you have any question and or suggestion, here are my email address. Welcome to contact with me. Thank you.
Yeah. Thank you very much for Tonia's presentation. It's always happy to see if we design a system and the system fits what customers need. Thank you, Tonia. Thank you, Tonia, again.
Thank you. Thank you.
Okay, so next one, we are glad to invite Tzu-Hung, who is the CEO of Lab Help. And today, Tzu-Hung will representing OncoRNA Lab to present the data. The topic is comparison of 3 different RNA isolation methods, which is FFPE material with regard to time, concentration, purity, and fragment analysis. Let's transfer the time to Tzu-Hung. Welcome.
Thanks,
Tzu-Hung.
Alex.
You're welcome.
Can you see my screen?
Yes, clear.
Great, thank you. Thanks for inviting me, Alex. And indeed, today I will present some results from iCatcher that were collected by the OncoRNA Lab from Professor Jo van der Zompelen in Ghent, Belgium. Like Alex said, my name is Jeroen Poodt. I'm the CEO of Lab Help, Lab Automation, and I will present. Oh, yes. A short presentation in 10 minutes. So, in fact, we are a middleware company, but since 2014, we also distributed and sold liquid handlers and also catch-gene products. And today I will present data from one of our customers, the OncoRNA Lab, Professor Jo van der Zompelen and Philippe de Kruijenaren. And they did a comparison between the iCatcher FFPE tissue RNA kit and 2 other methods. And they looked at the total time, the concentration, the purity, fragment analysis, and downstream next generation sequencing. So their test setup, they included 16 FFPE tumor specimens from diffuse large B-cell lymphomas. And they used 6 reference materials. And those 6 reference materials were 3 microsatellite stable cell lines and 3 microsatellite unstable cell lines captured in FFPE. Then they isolated using 3 different systems, 1 manually method and 2 semi-automatic methods. And they compared the time, the RNA yield, RNA integrity, and RNA purity, and did some additional NGS experiments. And they sequenced in 2 different methods. They used the RNA TrueSeq exome sequencing, so a library prep that uses amplification of the exome, and they used the smarter stranded total RNA sequencing kit that does not use amplification, but looks at the total RNA. So some results. So method A, B, and C are mentioned here. And method C is the iCatcher. Method A is the manual kit, and method B is another semi-automated kit. So looking at the total turnaround time, the manual method took the most time. Method B was 3 hours and 15 minutes. iCatcher took a bit longer, but could hold more samples. So if you look at the number of samples that you can process in one working day, iCatcher was the best. So 36 samples, FFPE samples in one day. If you look at the RNA concentration or the yield, they used 2 different methods for determining the yield: the nanodrop and the qubit. And if you look at the total yield, all methods are more or less the same. So the median is between 8 and 13 nanograms per ml. Here you can also see that they distinguish the results between the patient materials and the reference materials. If you looked at the purity, so they looked at the ratio 260-80 for detecting impurities with proteins, and they looked at the ratio 260-30 for impurities with regard to phenols and salts. And there, iCatcher was much better. So 260-80, it's more or less consistent. But if you look at the impurities with phenols and salts, the iCatcher gave the best results. So the most clean material. They also looked at fragment analysis, and from all patient materials and all reference materials, fragment plots were generated. And this is a representable plot from one of the samples. You can clearly see the iCatcher method in red, showing fragments over the entire range, from small to very large fragments of RNA. Whereas the competitors showed, well, some peaks at the lower part. So there were more small nucleotides of RNA present. So in general, more longer fragments present in the iCatcher RNA eluates. If we looked at, well, more or less the same, the RNA integrity, they used the percentage of fragments that were larger than 200 nucleotides. And here you can also see that iCatcher is outperforming method A and B. So in this case, the median showed that from all of the samples, 80% of the fragments is larger than 200 base pairs, nucleotides. And if you look at the statistical analysis of that, you can also see that it is statistical distinguished. So the iCatcher is the most right side. This is the these are the reference materials. These are the patient materials, and you can see that the iCatcher has higher fragments, and the percentage larger than 200 nucleotides is the biggest. At the end, they did a next generation sequencing on all these FFPE materials, and they showed 2 methods. So here you are looking at the uniquely mapped reads of the results, and the uniquely mapped reads is representative of the usability of your data. So if you look at the method exome sequencing, where there is amplification of your exome, you can see that the 3 different methods perform equally. So there is no real difference if you use amplification after isolation. If you look at the total RNA sequencing, then there is a difference. So you can see that there is at least a significant difference between method A and iCatcher. And from the 3 methods, iCatcher performs the best. So gives the best and the most usable data for further analysis. So to conclude, I made a short overview. If you look at the yield, there in their hands for FFPE RNA, all systems gave the same result. The yield was comparable. With regard to purity, iCatcher was better. If you look at the percentage above 200 nucleotides, iCatcher outperformed the two other systems, which much better. If you are talking about next generation sequencing and you look at exome sequencing with an amplification step, all three methods produced more or less equal results. If you looked at the uniquely mapped reads for total RNA sequencing, so without amplification of the exome, the iCatcher gave more uniquely mapped reads and so more usable data. That was the short presentation I had for the data of the Ghent laboratory. This data will be submitted for publication in 2 different papers in the next few weeks. So whenever these papers are out, I'm sure I will let Alex know, and he will transfer the publication to everybody who is interested. Thank you.
Okay, thank you very much for Tzu-Hung's presentation. And again, iCatcher was born for liquid biopsy, but we know liquid biopsy is the trend, but tissue biopsy is the golden standard. So very luckily, we not only can handle liquid biopsy, we also can handle iCatcher, also can handle tissue biopsy, especially for FFPE RNA. Actually, it's a very difficult sample because we know RNA already very difficult, but FFPE means very aged and waxing tissue RNA, which is very difficult to keep the integrity. And from the comparison, we can see the iCatcher really can capture more and more complete fragment of RNA. Thanks for Tzu-Hung. Okay, thanks for your sharing. Thanks again for all participants. And if everyone have any question, you can just contact us. And this is our website. And for this webinar, we have special thanks for our best support from Biomed in Singapore and from Central Taiwan Science Park, which is the very big science park in the central part of Taiwan, and genomics and the lab help automation company. Okay, thank you very much for your joining, and welcome to browse more on our website. Thank you for your joining. Thank you. Thank you. Thank you. More and more people turn on the video. Thank you.
Yeah.
Thanks, Alex.
Thank you. Thanks, everyone. Okay. Bye-bye.
Bye.
Okay, we got some new people turn on.
Yes, yes, great.
Thank you.
Thumbs up.
Thank you for your joining. Bye-bye.
Bye-bye.
Goodbye. Thank you.
Bye.